Our first speaker will be Bryony Forbes. So she's currently a professor in medical biochemistry. She's going to be talking about diabetes, and if the title's anything to go by, it's going to be very exciting. How can cone snails and platypus help us treat diabetes? Our second speaker will be Lido Papadopoulos, cancer, superbugs, and the gut microbiome. Lido is an infectious diseases physician and a clinical microbiologist. And then to bring us home, we've got Associate Professor Michael, who is a self-confessed gene junkie and chair of our Flinders Foundation Research Committee. So I just want to acknowledge Sylvia Wilkerson and Georgia Costaloo from the Flinders Medical Centre Volunteer Service here today. We've got a wonderful partnership with the Volunteer Service, and that enables us to fund research projects via scholarships. So sets PhD students on their way to stardom, and Lido's the beneficiary of some of that funding, and her work that you'll hear about today is really exciting as well. So, Bryony, over to you.
As Ross said, we work on cone snails and platypus. Now, what on earth could that help? How could that on earth help humans and treat disease? So, this actually is a picture of a cone snail. They live in oceans and they produce a venom. So I'll come back to that in a minute. The answer to my question, what on earth or what do we have in common with cone snails and platypus, is the hormone insulin. So in fact, humans and cone snail and platypus produce this hormone. So here you can see a human, and the highlighted is the pancreas where the hormone insulin is produced. So insulin's a little tiny protein. So proteins of, for instance, a protein is egg white, you might be familiar with. Well, it's a similar kind of molecule. But here in the body, it's produced in the pancreas, it circulates around, and its job is to keep our blood glucose at the right concentration. And glucose is a very important energy source. So all our cells in the body need energy to function. So if we have the right concentration of blood glucose in our circulation, then we'll be in a good situation. So what happens when we eat a meal is that we get insulin produced so that we can control the incoming glucose from the meal and control the levels. So here you can see over a 24-hour period, the blood glucose levels of insulin go very rapidly up after you eat the meal. And then there's a kind of a baseline insulin level that makes sure everything's smooth in between, the level of glucose is smooth in between. So glucose comes from our diet, comes from carbohydrates, so bread for instance, and carbohydrates, when you ingest them, are broken down into simple sugars like glucose. So insulin helps to take that glucose up into the circulation and deliver it to tissues and get the cells to take that in. So here we are. Insulin works on a cell. This is a diagram of what we think a cell looks like. It acts like a key. It binds to a lock on the outside of a cell. The key opens the lock, and then that leads to the opening of another gate on the outside of the cell to let glucose in. So without insulin, the cells can't take glucose in. So imagine if you're running a race, muscle needs a lot of energy. We need to open those gates and allow the glucose in. It's broken down to energy. It's used by the cell to drive muscle contraction, for instance. If we have plenty of glucose, the cells take it up and put it away for storage for later. So that's insulin's role in the body. What happens if we don't have insulin or it doesn't work properly? Well, we have diabetes. So there are two types of diabetes, type 1 diabetes where that pancreas can't produce the insulin, and another type which is the most prevalent form, which is type 2 diabetes. And here, the cells can't respond. So insulin can bind to that lock, but the cells don't get the message. So they don't open the gate for the glucose to come in. So what happens if we have diabetes? Basically, in both cases, glucose can't get into the cells that are needing it, muscle cells for instance, and and you have lots of glucose left in the blood. But you feel a bit hungry and tired because the cells don't have the energy to work. So that's one of the first symptoms of diabetes is feeling very hungry. In some situations, say if you had injected too much insulin when you were treating your diabetes, you might get what we call low blood glucose or hypoglycemia. And here, the glucose levels are so low that there's not enough to really supply the brain, and you get very clumsy and slow and confused. And that's important when we talk about cone snails, and I'll come back to in a minute. So, diabetes is a big problem. Lots of people in the world, almost 8% of the population of the world suffer diabetes. So it's an enormous problem. And it costs people personally very dearly, and also costs the health budget hugely. One of the major things that people don't realise is that in the long term there are all sorts of nasty long-term complications, such as diabetic ulcers, which ultimately lead to many amputations, which is terrible, problems with sight and so on, and also an increased risk in cancer. So we're very interested in understanding how, if you don't control diabetes properly, how that leads to cancer. Okay, that's the problem. How do the cone snails come in? Well, a colleague of mine in Texas did some gene sequencing, or actually the sequencing of the message of the genes. They sequenced the expressed genes in cone snails, in the venom actually of the cone snail, and compared that to the human sequences, and they found, lo and behold, that they had insulin in their venom, which was a big surprise. So how does that work? So can I do this? I just have a little movie here of a cone snail. So here's a cone snail trying to catch some prey. It turns out that some of these cone snails extrude their venom over the prey, the fish, and that leads to insulin being taken up by the fish, and they go all clumsy. I told you before, if you have too much glucose, too much insulin, it slows you down. So that means that the cone snail can grab hold of the fish and eat it. So that was really fascinating. We then looked at the insulin in a bit more detail and the chemical makeup of that insulin. And we found that it works very well in fish because it looks like the insulin in fish. So the cone snail has adapted its insulin very cleverly to be used in fish. It also has a particular part of its molecule missing that normally is very important to open that, to be binding to that lock and opening it. So that was a big surprise to us. How could it work without that particular part of the molecule? So we thought we've got something really new here, a new way of insulin working. And we used a very high-end technique called X-ray crystallography. It's like a large molecular microscope, very tiny detail, to look at the molecular structure or three-dimensional structure of that protein. And we could tell from that and predict that it binds to the lock in a different way. And that gave us the basis for designing a whole lot of new insulins with new properties. And in fact, since this time, so this was about a year and a half ago, we've discovered this. We've taken that information on to build new insulin molecules that are now being developed as new diabetes therapies. So that's where we're at with that work. So, what about platypus? Well, we thought maybe we could take the same approach with platypus as comparing the genes across the two species, because that was very successful in the cone snail. Platypus and echidna are monotremes. They are one of the first mammals to evolve. So they're like a snapshot back in time as to how mammals looked when they first evolved. And so we knew that they would have some interesting information in their genetics. And in fact, with this, my colleague Frank Bruxner at Adelaide Uni, who's a geneticist, we looked at the gene sequences again, and we found another molecule which controls insulin release that is different, and now we're developing that to be a potential type 2 diabetes. So I hope that you can see that the take-home message is that comparing genes and proteins from different species really gives us a new way to look at things, and then that allows us to build upon that information and make new treatments. So I just end up by mentioning all the people that have been involved, lots of people all over the world. I haven't got the international collaborators here, but I'd like to also point out and confirm that the Flinders Foundation money has indeed led to further funding. So we've had NHMRC funding starting this year, and Diabetes Australia funding for this project and related projects that bounce off the Flinders Foundation donations. So that's really fantastic. Thanks very much. That's a snapshot of our...
So I'm an infectious disease physician, and so one of my greatest worries is antibiotic resistance. So that's the ability of bacteria to become resistant to antibiotics. And this is actually one of the greatest problems that is facing medicine today. It's not just because if you get bacterial infection, you may not be able to treat it, but many advances in medicine, for example, organ transplantation, chemotherapy, surgical procedures, prosthesis, etc., depend on antibiotics being effective, because otherwise patients wouldn't survive these sorts of procedures and operations. But we do have a major problem. It's a greater overseas, but there are bacteria now that are developing resistance to almost every known antibiotic, and these are spreading, especially in hospital settings. So it is actually quite scary. In Europe, and especially in Greece, where my people come from, there's a very bad problem with a germ called CRE. It's resistant to carbapenem, that's the C. That's one of our last line effective antibiotics, and it's in this particular germ called Klebsiella, it's more than 50% are resistant to sort of the last line type antibiotics. So we have serious problems, and these, it's not just Greece, it's all of the Middle East, a lot of Asia as well, and coming on its way towards Australia as well. So these are actual problems right now, especially in a global setting. And these germs pose a particular threat to the immunosuppressed patients. Those are the patients whose immunity is down because they've had chemotherapy or transplantation. They're the ones that will be the most affected by these germs in hospitals when they get them. So if a patient who's immunosuppressed gets a bacteremia, so a bloodstream infection of, say, this germ, the mortality is about 70%. So it's very lethal disease. So I'm interested in how antibiotic resistance spreads and how this relates to the gut. So bacteria, especially the ones that carry those sorts of resistance, are actually exchange genetic information. In sort of a, you can see in the picture there, is a bacterium, sort of bacterial sex, where they exchange plasmids with antibacterial resistance. And in the gut is where all the bacteria get close together. So it's like a big party. They're all having a good time and exchanging genetic information, which includes resistance to antibiotics. So the gut is actually an important place where this happens. So now that we've got new technology, we're able to look in detail at gut microbes. So this couldn't happen before because many of these bacteria are very difficult to grow in culture. Some have never been successfully cultured. So the only way we can look at that is through sequencing. And this technology called next generation sequencing allows you to see all of the different microbes at the same time and see their genetic sequences. So what is happening to the gut for example, when you give chemotherapy and antibiotics. In a healthy microbiome, you have a large diversity of different germs. That's normal. And the type of bacteria is mainly these germs called firmicutes. They're anaerobes. They live happily in the gut. They're the good guys. What happens when you give antibiotics and chemo is that you have something called dysbiosis. The microbiome changes. What happens is you get less diversity. You've actually killed off a lot of the good bacteria in the gut. And instead they're replaced with other bacteria. For example, these blue ones are the proteobacteria. This is the type of bacteria that I'm most concerned about, the ones that carry the CRE type resistance. So you get a real expansion. So in the normal population, these are very low amounts, but then it expands to be much more after treatment. Another way to look at it is in patients. These are probably some of the sickest patients we have. They're the leukemia patients getting bone marrow transplantation. They're very immunosuppressed and they have a lot of antibiotics and chemo. What happens in their gut is that you pretty much wipe out almost every bacteria there, but one or two will survive. And they will take over the entire gut. And once that happens, once you have one type of bacteria dominating the entire gut, this leads to bloodstream infection. In about 7 days from this, a large majority get a bloodstream infection. About 50% of them can be predicted based on what is in the gut for a bloodstream infection. And these events are lethal in many of these patients, so it's a very serious problem. So part of what I did in my PhD is have a look at a model of what might be happening with chemotherapy and antibiotic resistance. So my theory is that it doesn't just take antibiotics to cause antibiotic resistance, but that chemotherapy by itself will do a similar thing and cause antibiotic resistance in the gut. So just as a guide, we'll look at good bacteria. These are the bad sort of bacteria that we don't like growing up. And these ones are the bad ones that are also resistant to antibiotics. So in the normal gut, you mainly have good guys, maybe a couple of these not so good ones floating around, and an intact gut mucosa. Once you have chemotherapy, it destroys a lot of the gut cells, and there's what's called mucositis. So this causes diarrhea in a lot of patients, and the gut wall lining can be leaky, and the bacteria can get into the bloodstream. But you also change what microbes are there, because the chemo itself will kill some of the bacteria. And a lot of the good bacteria will protect you from the bad ones taking over. So also if you're in hospital, you might be exposed to one of the environmental bad bacteria that has resistance to that stuff. Now chemo is a mutagen, so it will mutate DNA, and that's how it actually kills a lot of cancer cells. But it doesn't mean it won't mutate the DNA in bacteria as well. So some bacteria will be mutated by chemotherapy. This actually turns on an SOS help me response, and they mutate at a much higher rate when they're activated this way. So they will become resistant to some antibiotics just based on this mutation. So now what happens is you've got chemotherapy has changed what's in your gut, you're exposed to a hospital environment, and you've got new mutations. These bacteria now have an opportunity to multiply and take over your gut. And also other non-bacterial things like yeast will start growing in your gut as well. And the last thing is, because of the damage to your gut, one of these guys will go through the lining, the barrier that's normally there, and cause a bloodstream infection. That is the real point where it's dangerous. Now, most bloodstream infections are treated quite successfully by antibiotics, The problem is when this germ is not sensitive to the antibiotic that we're using. So if we're using the wrong antibiotics, that bloodstream infection can be a late bloomer. So we really depend on the right antibiotics working in our patients before we even know what's wrong with them. So what is, solution to some of these problems of resistance? Infection control has been, tried, so that's where we isolate patients, use gloves, gowns. Of course, this is helpful, but it's not actually contained in a lot of these outbreaks. These germs continue to spread around the world despite infection control procedures. New antibiotics, desperately needed, but because a lot of it has to do with economics, there's not much money in making new antibiotics because they'll just be reserved on the shelf, so the drug companies don't feel like investing in it. So there's a huge shortage of new antibiotics, which is going to be a big problem. In my side of the story is looking at the microbiome, so the gut microbes, and seeing how this could help us prevent this colonization with resistant germs and help prevent infection, essentially. Mainly by using sort of fecal transplant or probiotics transplant, that is. So fecal transplant is where you take the poo of a healthy person or a healthy mouse in this case, and replace the bad germs with the good ones that way. So with mice, it's really easy to do because they just eat each other's poo. So you just give them a nice poo to eat, and it works. So this mouse has had antibiotics and been colonized with a superbug called BRE, and the other one is the Klebsiella that we looked at earlier, 2 superbugs, and the gut is full of these superbugs. But when they get the new, the fecal transplant, these go down to very low levels. So it seems to work in eradicating these superbugs from the gut in the mouse. What about humans? There's case studies that shows work in individuals. So this is a person who was colonized with all of these superbugs, so there's lots of them, because he was living in the intensive care unit in America. And then he got a transplant here, and the number of superbugs he was growing was really reduced a lot, and only to basically MRSA, which is a skin germ, so it wouldn't be eradicated by a stool transplant anyway. Then he got it back here, and then he died. So it just shows you that it is possible to use this technique to, at least in some people, to eradicate superbug carriage. So my PhD is based at Flinders Foundation Research, so that's over in the main building on the 5th floor. I'm supervised by Geraint Rogers, Prof. Dave Gordon, Prof. Steve Wessling. And what we're doing is, as I explained, we're looking at antibiotic resistance in patients getting chemotherapy. So I'm currently collecting stool samples from patients who are just about to undergo chemo for the first time, and just seeing what happens in their guts, just to show what is happening with just chemotherapy, because a lot of people haven't looked at this. They've looked at it in combination with antibiotics and not just chemo alone. So there's surprisingly little research into what exactly is going on with just chemo in the gut. And also look at if this is causing mutations to the bacteria, as I would predict. And this is affectionately known as the poo study upstairs in the chemo suite. And then I'm also doing a trial in patients who have resistant infections that are coming from the gut, so recurrent ones. This is quite a problem for these patients. We currently get infections, they get antibiotics, and then they get infected again, and it's a cycle that's very difficult to break. And we're going to see if the fecal transplant from a healthy donor can stop that cycle. And we're currently recruiting patients for that trial as well. And another thing I've been working on is understanding what microbes are in the fecal transplants, how we process them, and what is dead and alive in these transplants. And this will help us understand why this therapy works. What bugs are actually going into people and which ones are working too. And this will help us provide a therapeutic that is not from just human poo. So we will try to develop therapeutics that are specific bacteria in a mix that are much safer to deliver than other people's stool samples, which, you know, you can understand comes with some risk, even though we screen them even more than a transplant would screen. But it's always a slightly risky thing to get people screened. And collaborating with others, especially in the cancer space, to develop trials and observational studies looking at the microbiome in this area. So thank you very much. This is a Flinders Foundation for PhD, so thank you very much for this research.
Good morning. Thank you all for coming. It's great that Bryony and Lito spoke before me because they've introduced quite a few of the subjects that I'm going to talk about with my talk next, which is actually on bowel cancer. We do a lot of work upstairs, so I've got a lab upstairs in this building. We do a lot of work on bowel cancer. And I'm going to talk about what it is about bowel cancer cells that makes them different to normal cells in the way they produce energy and the way they're influenced by things by your diet. So this is going to come back to what Bryony's been telling you about insulin and diabetes and energy production. And it's also going to be talking about the microbiome and the bacteria in your gut that actually feed your bowel cells that Lito's been introduced. But first, I need to give you a very brief introduction to molecular biology. So molecular biology 101 in 2 slides, okay? Don't get spooked. So essentially what happens in a cell when all of the cells in your body have the same DNA, right? On the chromosomes, half from your mum, half from your dad. But what makes one cell different to another, a liver cell to a brain cell, is that a different set of genes is active in one compared to the other. You need all the things in a muscle cell to be present, like all the fibres that pull the muscle. You don't need those in a brain cell, you just need to be sending out little electrical shocks all the time, right? So different genes do that because the different genes, when they become active, produce different proteins. And Brian showed you some really nice pictures of the insulin protein before. Now, as she said, you get different genes that are active, and when a gene becomes active, it rewrites itself into a little message, which is a copy of that DNA sequence, that comes out of the nucleus of the cell into the cytoplasm, and that produces the information that these little protein factories called ribosomes need to link the little amino acids together to make that protein. That's all you need to know about that for now. So this is the dog: DNA, RNA, proteins. And that's growth. And what happens in cancer is that it's associated with changes in this. So the DNA can get mutated, and so you'll get change in the sequence of the RNA, which leads to a change in the amino acids that go into a protein, and that might make it a more aggressive protein that might lead to a cancer. Or it might take away a protective protein. Similarly, you have epigenetic factors. So you have factors that change which of the genes are active or not active. So which RNAs get produced in a cell that maybe shouldn't be produced in that cell. So they're the 2 major things to worry about. Now what we work on are a different set of genes that produce RNAs, but these RNAs don't go on to produce protein at all. They get processed into these little snippets, and they're called microRNAs. And what they do is they actually attach to a target RNA and stop it from producing the protein. So they shut down the protein production for a specific target gene, okay? That's kind of what you need to know. So I've introduced you to microRNAs. Just remember, when a microRNA goes up, its target protein or the target RNA goes down. Right? We're going to come back to it, and there's a test here. Okay, our genome contains 2,500 or a bit over that microRNA sequences, lots of them. 2/3 of all of our genes are regulated by microRNAs, at least 2/3, probably more. And they regulate almost all the developmental and metabolic processes that go on in our body. They can play roles in promoting cancers or protecting you against cancers. Now I'm going to switch to metabolism. Okay, so metabolism is how your cells, how you make stuff and break stuff down. How you produce energy to make stuff, and how your body breaks stuff down to get some energy back sometimes, okay, essentially. But I'm not going to worry about you and your diet so much, but I'm going to worry about your diet and how that affects the bacteria in your gut, which is what Lita was talking about before. So you all know where your colon is now, I'm sure. And Lita's told you that the lining of your colon is lined with bacteria, and that's what they look like. And you only really take notice of it when the bacteria are nasty. But I want to tell you about a lot of the bacteria that are good, and Lito told you that you've got a lot of good bacteria in there as well. And I want to focus on some particular ones that produce this compound called butyrate. So they break down the fibre that you eat to produce this stuff called butyrate. Now, you all know that you should be eating a high fibre diet, don't you? You all know that, don't you? And tell me why. Why should you be eating a high fibre diet? What's it about? How's it work?
Run marathons and half marathons.
You can run marathons and half marathons. You can. How does the fibre affect you though? Why has it killed you?
Well, I've been involved with bowel cancer for 44 years.
Yep.
And that's probably what's helped me. And is Scotland still, mum's side of the family come from Scotland in 1846. Are they still fourth line in the world just for bowel cancer? Because if anyone's got Scottish heritage, make sure you get checked.
Yep. And the diet's not so good because there's not a lot of fibre in a Scottish diet, unfortunately. That's a big problem. And seriously, it's a problem.
Well, I've run marathons and half marathons for
Yeah.
2.5 years.
So most people will tell you the reason you should have a lot of fibre in your diet is because it just moves stuff through the system, right? You know, you eat fibre, it all works very nicely, just move it all through. It's only part of the story. The main part, we think, is actually because when you have fibre in there and it helps build up these bacteria because they get lots of food because they like chewing on the fibre and they produce a lot of this stuff called butyrate. Butyrate comes from the Greek word butyros, which is Greek for butter. You eat butter, 4% butyrate. Often doesn't smell that good. Parmesan cheese, that smell, butyrate. But the bacteria are producing that in your gut, and that is the preferred energy source for a normal colon cell. They metabolize it faster than glucose and sugar. They burn it really quickly. But if a cancer cell gets exposed to it, it dies. It kills cancer cells really. Okay? Let me show you an example of that. Okay? So it kills normal colonocytes, normal bowel cells, kills cancer cells. Could you ask for anything better than that? Now, the work I'm about to show you is done by this young lady here, Karen Humphries. She did a PhD in our lab a few years ago, stayed on and did a postdoc, has done some really nice work. Karen was a dietitian actually, who, having done a medical science degree, then dietetics, decided to do a PhD, 2 years postdoc, and now she's gone back and she's doing medicine, second year. Professional student. Anyway, so she showed, so here are the colon cells growing in culture in red at the top. If you add a little bit of butyrate, you slow them down a bit, 5 or 10 millimolar of butyrate, you stop them dead, right? They stop growing. And these are the sorts of levels that are in your bowel if you eat a good high fibre diet with a lot of resistant starch. Okay? So the take-home message is butyrate protects you against bowel cancers. So Karen was asking the question, what is the difference between a normal cell and a cancer cell? Why do they respond to butyrate so differently? And she was interested in the fact that cancer cells have different microRNAs to normal cells and wanted to test whether those microRNAs might be the cause. So one of the first experiments she did, and I'll explain this to you, is she took cancer cells in a dish and exposed them to butyrate, and then looked to see which microRNAs go up or down. And it's one of these sequencing type experiments that Lita was talking about before. And this is a plot that essentially tells you all the ones that are out this way are the ones that get turned on, and all the ones that come out this way are the ones that get turned off by the butyrate. And we were interested in a lot of these microRNAs because we knew that they're associated with cancer. They're an oncogenic cluster of microRNAs that are often turned on in bowel cancer, and we showed that they're repressed by butyrate. And she actually showed that in fact, these microRNAs are directly responsible for the different effect that butyrate had. Because if you treat with butyrate but add in a lot of synthetic microRNAs into the cell, you prevent that cell from dying. Okay, so the microRNAs are involved in this process. And without complicating too many things, a microRNA is only ever as good as the target that it regulates. Remember, microRNA goes up, the target goes down. Okay? So in this case, these cancer-associated microRNAs go up. The proteins, the genes that they regulate, are protective ones, they're tumour suppressors. They go down. You don't want them down, you want them back up, right? And so we showed that when you treat with butyrate, so this is the amount of protein, these are the protective proteins. So where butyrate was present, you see these getting turned back up as the microRNA gets turned off. We also found other microRNAs that can kill the cancer cells that are affected by butyrate. So I only want you to worry about these pictures here. So here are the cells growing in culture. I hope you can see that. Right? If we add one of these microRNAs, MIR15, and infuse the cancer cells with MIR15, you can see we can knock them off pretty easily. So we're trying to develop some therapies around exploiting these microRNAs to deliver them therapeutically to a cancer cell to knock it off. The hard part, of course, is getting them into the cancer cell. And we're doing some quirky stuff around that at the moment. Okay, so I've told you all about cells in a dish and what's so exciting about butyrate cells in a dish. We took that one step further. So Karen, gung-ho dietitian, she was part of a large clinical trial that we did with CSIRO. And they were interested in this product that they have called Star Plus. So that's actually a resistant starch, which is a type of fibre, and it's the starch. So resistant starch, it actually comes from maize, from corn. And it's the stuff they put in Wonder White bread. So you buy all this white bread and they tell you it's actually high fibre bread, even though you know it's processed white bread. The reason they can say it's high fibre is because they've added all of this maize starch into it. And it's actually good. What CSIRO did is they took this maize starch and they added a bit of butyrate to it. So they've actually stuck bits of butyrate to the maize starch. So even before, so that gets down to your bowel, and even before your bacteria have to ferment the fibre, you already get butyrate because they've added it in. Okay, so it was testing this Star Plus stuff, this butyrated starch. But it was testing it in a background of a lot of red meat. So the study was like this, crossover study. 4 weeks, so we had 25 volunteers and 23 saw it through. A controlled diet for 4 weeks. Then we gave them 300 grams of red meat a day on top of what they would normally eat. A washout for 4 weeks, and then red meat plus the resistant starch, plus Star Plus. Okay. So we're asking the question, what happens if you eat a lot of red meat, which is known to predispose you to bowel cancer? And what happens if you add this high fibre, this resistant starch, when you eat the red meat? And they did all sorts of things from these poor patients. They collected their urine, their blood, their stools. We took rectal biopsies of them, we swabbed them, we did all sorts of things. And the take-home message was that we were looking at those cancer-associated microRNAs that I told you about before. And if patients just ate the red meat, the levels of those microRNAs in the rectum, the lining of their rectum, we took little biopsies, went up about 30-35%. But if they had the resistant starch as well, back to baseline, which is good. Now, I'm not telling you that those microRNAs going up in and of themselves causing cancer. We don't think they do. But remember, these buffer against other genes. So if you've got other mutations that are occurring in those cells at the same time that can lead to cancer, these microRNAs aren't going to protect you or are going to exacerbate a problem that's already there. So anyway, so that turned out to be a really good news story. The story is, enjoy your barbecue, but eat the potato salad. No, sure. Potato salad is really good in resistant starch. So starchy foods that get cooked, heated, and then cooled down, the starch granules actually condense into these resistant starch molecules that are really protective. So for example, cold rice, sushi, right? It's really protective. But also just normal high fibre diet, eat the salad. Okay, so we're actually doing similar work that we're just about to start. So there's a new clinical trial that's going on around the country. And these are people who have a genetic predisposition to bowel cancer. They've got familial adenomatous polyposis, FAP. And these poor people have inherited a condition where generally teenagers, their colon looks like velvet because it's lined. In the extreme case, it just looks like polyps, it's a carpal polyp. And they will likely have bowel cancer by the time they're in their mid-20s. And so often, these poor kids have to have their colons removed. You can imagine the effect that has on you as a 15, 16-year-old. So anyway, they have, even if they have their colon removed, they have to have it stitched back, and because of the way surgery happens, you still always have a bit of that lining that's predisposed to bowel cancer. And so we're trying to see if giving them an added butyrate in their diet reduces their polyp burden. And Star Plus is good for that, this stuff, because again, because they've got a very different bowel, they have different bacteria in there as well, right? So they haven't got as many of the fibre-chewing bacteria. So this Star Plus stuff not only gives you fibre, it gives you the butyrate anyway. So we're hoping that will help them. And we're in the process now of testing how the butyrate influences all of their genes and their microRNAs, and particularly what's happening in these pre-cancerous polyps. That's gold dust for us. So that's what we're working on at the moment. But this all comes back to metabolism. So one of the proposals for why a normal cell responds to butyrate differently to a cancer cell, we've shown you that it's microRNAs, they play a role. But another group has proposed that it's in fact a different metabolism. So a normal cell burns the butyrate really quickly. But cancer cells make their energy differently. And for that reason, they've proposed that they don't burn that butyrate quickly enough. It gets a chance to build up, and that way it influences the way your genes are expressed in the cells. And that leads to the death of cancer cells. That's what they've proposed. And that's kind of interesting to us, because a lot of the microRNAs that we've been looking at, we think are associated with energy production anyway, right? So we're trying to tie all this together. Butyrate, metabolism. There are a couple of ways that cells can make energy. Well, there are lots of ways that cells make energy. The main ones are the mitochondria. So they're the energy-producing factories of the cell. And we wanted to see, well, if we could go in and just find out which microRNAs regulate energy. But if we do that, and we shut down one part of the genes in one part, then the other part pops up and starts producing energy in the cell, right? So cells are damaged. So we thought, how are we going to get around this? We thought, why don't we try shutting down the mitochondria and then mess with this other part, energy production part, which is the main part that everyone thinks is involved in cancer cells. And this is the part that's used by cells when they run out of oxygen. You know, when people, you're a marathon runner.
Used to be.
Okay. So when you're running a marathon, or actually more importantly, when you're sprinting, and you can only provide so much oxygen to the cells in your legs, and they start building up lactic acid. You've all heard of that probably, lactic acid. Lactic acid is a byproduct of this pathway. Because what they're doing is there's not enough energy, they have to produce, there's not enough oxygen, they have to produce their energy quickly, and so they use this pathway and it produces lactic acid. So we decided we were going to try and shut this down and just look at this cancer-associated low oxygen pathway. And the way we did that was with a drug called Metformin. This is where it comes back to diabetes. So many of you are probably familiar with Metformin. Some of you may be taking Metformin. It's produced from French lilac. It's one of the most prescribed drugs in the world. More people use insulin than for type 1, but for type 2 diabetes, it's the first line treatment. And it works by essentially shutting down the mitochondrial energy production. And in that way, it prevents the liver from producing lots of glucose that it releases into the blood to have the effects that Brian told you about. And it turns out that people who take metformin, compared to other diabetics, have a lower incidence of cancer. They also, for what it's worth, have a lower incidence of cardiovascular disease, have a lower incidence of neurodegenerative diseases. Metformin actually looks like pretty good stuff. So we were interested to see how metformin works, how might it protect you against cancer, and we wanted to shut down the mitochondria. So I have this wonderful PhD student at the moment, she's in the front here, Ayla Aran. So Ayla won the university's 3-minute thesis competition last year, and here she was with all the other contestants getting that prize last year. So what Ayla did was she took bowel cancer cells in a dish and she treated them with metformin. She then measured which genes get turned on or off and which microRNAs get turned on or off. And she was looking at the patterns where you have a microRNA going down and its target gene going up, or the opposite, where the microRNA goes up and the target gene goes down. Because we think that's going to be informative in how the cells are responding to metformin at this gene level. And this is what the data look like. We call that a hairball. But you can tease into these data. So this is the new wave of genetic analysis. It's called systems biology. It's taking as many of these big data sets, gene sets, microRNA, proteins, metabolites, as much data as you can, ram it all into a computer, and get the computer to spit out a diagram for you, and it looks like that. But when you tease into it a little bit, you can actually start to see the pathway starting to emerge. So all of these, you can't really read them there, I guess, but all of these are genes and the proteins they produce, and the ones in black are the microRNAs that regulate them. And we can tell which of these proteins or genes interact with each other. And we know which ones are associated with things like cancer, like adhesion of cells, so when they want to clump together, development, responses to low oxygen, all these things start to emerge from these data. And we can look and get a bit of a snapshot. So what's happening in these cells? Well, the first thing is the major thing is it looks like they're responding to low oxygen. But that's because their whole metabolism's been changed, right? It's been swung away from that mitochondria to the low oxygen pathway. And all these other pathways, and we've chosen to focus in on this one because it looks like that pathway might be a good one for a complementary drug that we might be able to team up with metformin to knock off cancer. And you can go down into it in a little bit more detail. So here's just that pathway, and we can use that to identify which are the proteins we should pick out to design drugs against, to act with metformin to knock off cancer. So that's kind of where we're at. These data only came to me from Ayla 2 weeks ago. And so what we do is we now generate a hypothesis based on these data to tell us that protein looks really good. Many of you all know that protein. That's the BRCA1 gene that people get tested for predisposition for breast cancer, for example. So these genes make sense as targets, right? So that's where we're at the moment. Okay, the other person who's being funded by money from the foundation for us is Anne-Sophie. So Anne-Sophie is a surgical registrar from Germany, who for a year has put down her scalpel and come over to pick up a pet and learn molecular biology. She wants to know what's happening. In fact, she's spending a lot of the time on a bike. So here she is in the middle, a few weeks ago raising money to the cure and for the foundation and for the research that we're doing. So she's having a great time out here. And what Sophie's doing is she's combining metformin with libraries of all different sorts of drugs upstairs to see which combinations of drug and metformin are most effective in killing the cancer cell. And she's using, I don't know if many of you have seen it, but we have a robotic facility upstairs that does that, because to do these experiments, we can look at you know, 5, 10, maybe 50 drugs, doing it manually at a time. But if you've got a robot, you can do thousands and tens of thousands at a time. And we can scale up these experiments and do them meaningfully. And so that's what's happening at the moment. Okay, I'll leave you with that. Thank the folk who are in the lab. So Karen, Arlo, I've talked about, and Sophie, who's joined us recently. Mari keeps the lab running. We work really closely with Pam Sykes and Ganesh and Kitchener-Darcy, who have probably talked to you before, I think, here. And Lynn Tran, Pam's PhD student. He's previously done work with Metformin. Amanda, a lawyer, runs that robotic facility that I showed you, and we've got a fantastic team who do all the genomics analysis that's upstairs here in the Cancer Centre as well. And of course, we are so indebted to the Foundation for their support. The way they operate really well with Tour de Cure and that new FAP study for those patients with the bowel cancer predisposition, that's funded by from the UK at the moment. So thank you very much.
I'll have the first question, because I can. And just ask you, Michael, how important is seed funding for the work that you and your colleagues do?
Seed funding of, you know, 20 grand just makes all the difference. That lets you do the experiments, keeps your PhD students going, and you can generate those data. And the investment is really good. We had a look at that a while ago, and from the foundation, I think for every dollar that it invested, $7 came back in funding from larger funding bodies, so that was really good.
Any questions for any of the three speakers?
I want to ask, what happens when people have an intolerance to fibre? Because people with irritable bowel can't take such a lot amount of fibre.
They will do it. So the resistant starch might not be such a problem for you. So inulin and those sorts of. I think you need to do this with the advice of a dietitian or with your GP, but rather than just having roughage go through, maybe some resistant starches aren't so bad. The cold potato salad, cold rice, maybe not too bad because that's more starch than harsh fibre like in a husky type thing. So think about that as an alternative if you can tolerate it. If gluten's a problem, none of those are an issue, so you're okay. But it's all around also keeping your microbiome healthy. So when you eat, think about what you like eating, but also think about what those bugs in your gut like to metabolize. Because they look after you, they really do.
What's the role of probiotics? You know, the Yakult, all those sorts of things.
Yeah, I'm going to leave.
Well, I mean, theoretically, it should be helpful, but I don't think there's any strong evidence that they actually get down to where they're needed or really influence any health outcomes. Perhaps the only exception is in very little babies. I think there's been some shown in probiotics, but I think once you're older, it just doesn't seem to be working at the moment. But that's where we're trying to overcome problems with how they're taken orally. Maybe they have to come up the other side, and probably different types of microbes and what people are giving you in those tablets will be more effective.
Yeah. So your stomach does a really good job of killing off the bacteria that are in your limbs, which is why a lot of what's in your intestines tends to replicate what your mother had.
Yeah.
Right? So, well, what your mother had. So when, by normal delivery, that's a very messy process, right? And on the way through, you end up taking in a whole lot of bacteria from your mum.
Turns out that it was intentionally designed like that.
It was. And kids from cesareans don't have that infection, so they often have to get it otherwise. They'll get it from, actually you can profile the kids and they'll actually have more the breast skin bacteria represented in the gut than the kids who have had a cesarean.
The other factor is a lot of the bacteria that produce the butyrate and other helpful metabolites die very easily, so they're very sensitive to oxygen. They like anaerobic environments, and so it's really hard to get them into you without killing them first.
Another question,
sorry, more research needs to be done into that, but certainly in the pediatric population that have had the leukemias, they seem to still have what's called a dysbiosis, an abnormal sort of profile in their gut bacteria many years later. Of course, they've had much more intense antibiotics, probably than a normal sort of solid or a cancer tumor would have. But we do know in that population it does seem to be a permanent change. Also, I mean, just taking antibiotics is a huge thing. So people who are taking antibiotics quite frequently probably have a different, less diverse gut microbes than other people.
Do you think that can be counteracted by a particular diet at the time of having the antibiotics? So say if you've got somebody who's on a course of antibiotics for a 6-week, quite a long course, could they be helping themselves by changing their diet?
I think at the time you're taking antibiotics is probably not that helpful because they'll still kill the organisms regardless of what food you're eating. Certainly after you've finished your antibiotic course, it's very helpful to eat, which is good for you at all times. But, certainly would be even more helpful at that time to eat a high fiber or healthy diet. The problem is we don't really know if the antibiotics have completely eliminated a particular strain of bacteria. No matter what you eat, it's not necessarily going to come back, and that's where our therapeutics using live bacteria might be helpful in some way.
But in a normal gut, you can really dramatically change your the gut bug population with diet. So if you go from a carnivorous diet to a vegetarian diet, within 5 days, they've shown dramatic swing in the bacterial populations in your gut, totally different bacteria. So they respond to your diet really quickly. They even respond over the course of the day as you have different meals. You have a really sugary breakfast, that'll affect them one way. In the evening, if you have a lot of fibre, it'll affect them another way. So you get really quick responses to your diet, which is why diet is so important.
We used to, if people are on antibiotics, we used to tell them to eat yogurt because that would counteract the gut problem. Is that still the thing?
Again, that's very similar to eating a probiotic. So, I mean, it does have a few beneficial bacteria in it, but whether that's really making a major difference to your gut population, I would be doubtful that it's making a major difference. I think yogurt, I love yogurt too. It seems to, be nice that your gut makes you feel good. We don't really know the answer to a lot of these questions of how much your diet is extremely better. A lot of these research is just showing what we've always known, which is a healthy, high fiber diet is good for you. It's just explaining why that's good for you. But it's the same with anything. Just eating a very healthy, high fiber diet is very good for you. We don't, as I said, we don't really understand antibiotics and chemo, how much damage is permanent, how much can be modified by the diet. I think that's an area that still needs to be explored.
We were told diets, eating potato chips and potatoes wasn't good for your diet and would help you put on weight. Now it turns out I get stuck into all the potato chips I want.
So no, potato chips probably not. But potato salad, yeah, go for the potato salad. I love it, but there's not a lot of starch left in
the chips are also high fat and high salt, not great. So I think that you have to, I would say, I'm not a nutritionist, but I think everything in moderation is a really good way to go.
Okay, thanks. Just want to thank Michael, Bryony and Lido for joining us here today. That's great. Just a few things I just want to bring you up to speed on. One is the Karen Fitzgerald Lunch Club, June the 22nd. So raising funds for abused and neglected children that receive care here at Flinders Medical Centre. We'd love for you to be able to attend. If you haven't visited our website, it is relatively new too, so it's just to go on there, have a look around. You can purchase tickets to find out more information about that event. and also the Pink Yellow Blue Ball this year, which will be held on the 20th of October.
Which is lots of fun.
Lots of fun. Michael won the car last year, which is why he's saying that. That's why he has to do these talks, he can't get out. I forgot to mention earlier, this is what our new newsletter looks like, and they weren't on the chairs, sorry, but there is a pile of them up the back, so if you want to get a copy before you leave, please do so. Entertainment books, if anyone wants an entertainment book, you can ask our team, but also go onto our website and find out all about it. We've also got the City Debate coming up, Terry and others.
I wish I could still do
Briggsy will be
Well, my
here.
time- My time now would be anything like it was back in the early 80s, 40 minutes, 11 seconds.
If you know anyone that might be interested, or even if they're walking it, you can still raise funds for the foundation by taking part in the City Debate. And Dry July's coming up, so we didn't really talk about alcohol through that. Talked a lot about food and so. But one thing you can do, obviously, is go dry in July and raise funds. We've been great partners with the Dry July Foundation, and we're raising funds for a cancer wellness centre here in the Flinders Centre for Innovation in Cancer. So if you want to go dry, do so. That's it from me. Next forum date will be on Monday the 4th of June. And we're thinking at this stage the topic's going to be mental health, so please join us for that one. We'll be sending you out some information about it. That's it.